Hi all, welcome to this lecture of Agrobacterium mediated plant transformation, as I have seen in the Google Classroom, and I have been informed by Gupta sir that he has covered a lot of sections on the plant tissue culture side and the other aspects related to plant biotechnology. So this particular topic of plant transformation, which includes direct gene transfer as well as agrobacterium mediated plant transformation and its applications. I mean, a brief overview of application of transgenic plants. This we will be covering in this particular under this particular topic. Okay. So we'll start. All right. I hope you guys can see this. This one. So, as I said, it will be about agrobacterium mediated plant transformation, and there are some slides later down regarding the direct gene transfer. So, I'll first start with agrobacterium mediated plant transformation. So, as we will move along, we'll realize that this is one of the most commonly used plant transformation technique in dicots. And before I start telling about this, I think I should step back into the history that this was not originally was a technique for doing plant transformation. It was initially identified as the ability of agrobacterium to infect plant, create tumor-like things on the plant surface, and that was. And then when people started studying, I mean, this is what they have seen first, that there's a tumor-like thing present onto the surface of the plant. And when they started studying this, they realized that this is being caused by Agrobacterium tumefaciens, which has the ability of transferring a part of its genome into the plant genome. So this was, I mean, this was actually a virulence part of the, so it's a virulent bacteria. So, and it was part of the virulence strategy, where a part of the bacterial genome is being transferred into the plant genome, and then that transferred genome is being utilized for providing nutrition to the growing bacterium. So it actually started from here as a virulent bacteria. So you just imagine or just look at the beauty of biotechnology or plant biotechnology, that how naturally existing virulence technique have been converted into such a potential tool for plant biotechnology. So just start getting into the details about it. So genetic engineering, of course, when you talk about plant biotechnology or agriculture biotechnology, you talk about different tissue culture techniques and all of the techniques that are being used to improve the, improve the quality of, or improve the productivity or sustainability or quality of the crop that are growing actually. Of course, genetic engineering of plants is an integral part of it. So as it stands, that genetic engineering covers that DNA should get inside the cell, and for the DNA to get inside the cell, there are two possible ways. One is transient and one is a stable expression. Now transient expression means that it is expressed for a very short duration, and both of them we'll talk about, but just to give you a brief idea, that transient expression means it is expressed for a very short duration, and in this case the integration is not happening into the germ cells like the pollen grains or to the mother or to the ovule part. So it's not happening in there, the polar nuclei, not the polar nuclei, the egg cell present over there. So since it is not going through the germ line, it is not a stable transgenic. And on the contrary, that's why it is known as a transient, is only expressed for a very short period of time. And on the contrary, we have a stable expression in which the DNA gets integrated into the genome and it passes through the germ line, so it moves from one generation to another. And of course, agrobacterium mediated gene transfer comes under that, and so is the direct gene transfer. So, and then once inside the plant, that it also depends that how it should be expressed or where it will be expressed, all these things. So that involves the discussion about promoters and reporter genes. We will have, I have some slides for that as well. Now, transient expression, this is just an example of transient expression in plants. and Nicotiana benthamiana, which is belonging to the same genus as tobacco actually, which is Nicotiana tabacum, belonging to the same genus as tobacco. So Nicotiana benthamiana is a very commonly used for plant for transient expression. Here also we use Agrobacterium, but in this case it does not, we infiltrate leaves, the Agrobacterium cultures inside, so it does not go through the germ line, so it does not lead to the formation of stable transgenics. So what happens, say, I mean, we have specific purposes for using or specific needs for using this particular experiment. Of course, in cases where we just want to check where the particular protein is localized or whether the two proteins are interacting, we don't have to, we might or might not have to generate transgenic plants. Of course, we can do that. Nobody is stopping us, but we might not have to. So what we do, we just take Agrobacterium cultures containing two different clones, one expressing in place of protein-protein interaction. Or in case of localization, we can just use one Agrobacterium culture with one clone actually. And then we mix the two, incubate them, and then infiltrate them on the lower surface of the leaf. And 3 days later, as I said, it is only expressed for a short period of time. So it won't express during the first 24 hours. In the next 24 hours, on 48 day, you might see, you might start seeing a very small fraction, but the best expression takes place at 72 hours. And then beyond that also, you don't see much expression happening. So after 72 hours, we just take the sample for localization, or we do immunoprecipitation if you want to check protein-protein interaction. But later on, it is useless. So that's why it is known as transient expression. Now coming to the stable expression, which involves Agrobacterium-mediated gene transfer. Now, as I said, this is one of the most commonly used method for engineering dicots, but it's also used in monocots. It has been pioneered by J. Shell. And giving you some basic background about Agrobacterium, it is a gram-negative soil bacterium, and it is related to Rhizobia. And there are different species that are being that are functional, that are being used for research purposes. Some of them. One is tumefaciens, which we have been talking about, which causes the crown gall disease in many dicots. Then we have Agrobacterium ruby, which causes small galls on few dicots. Then another important one called rhizogenes, Agrobacterium rhizogenes, which causes the hairy root disease. This again is very widely used in some of the research where we need like good hairy roots. So people have started using Agrobacterium rhizogenes over there. And then Radiobacter, which is an avirulent strain. Now this is how a crown gall looks like, and this is how it is started happening actually. Or the signs started happening when people started seeing these tumor-like things on the plant surface. So once it requires a wound infection, it requires a wound. So infection actually takes place. The Agrobacterium infection actually takes place at wound sites. So whenever there is a wound on the plant surface, especially in the roots, it will start synthesizing a chemical compound called acetosyringone. Now, acetosyringone involves chemotactic responses from the bacterium. Once the acetosyringone is synthesized, it will attract bacteria towards itself. And this attraction, and then once the bacteria is present in the, in the rhizosphere, it enters the plant cell and then it can move. It can move through, like it gets entered through the wound itself, and then it can move actually, and that's why you see a crown gall being formed at the trunk part of the tree, whereas the bacteria would have entered through the rhizosphere, or is present in the rhizosphere. Now these galls are real tumors, and they can be removed and will still grow indefinitely. So this is an example of a real tumor that even if you cut out, it keeps growing. It indicates that all the genes, all the required ingredients that are required, all the things that are required for it to grow is already present inside this tumorous cells, inside this tumor. And that's why they are named as, or they are called as real tumors. So we'll see how it happens actually. Now, coming to like what causes the crown gall and how is crown gall a real tumor and can grow on its own. There is a Ti plasmid present inside the Agrobacterium. So what people have found actually, and how they started, what led to the discovery of Ti plasmid is people found that there is an extra chromosomal element that can move from a virulent strain to an avirulent strain and vice versa. So a virulent strain is getting avirulent and an avirulent strain is getting virulent, and that suggested there is an extra chromosomal element that is moving from one strain to another. And this particular extra chromosomal thing was found to be a large plasmid known as the Thai plasmid, which is also known as tumor inducing plasmid. So that tumor formation that we were talking about in the previous slide here is actually because of the Thai plasmids. Until and unless a Thai plasmid is present inside the agrobacterium, it is not going to cause the tumor. That's why this Thai plasmid is known as and this plasmid is known as the Thai plasmid or tumor inducing plasmid. Now, this particular tumor-inducing plasmid has a region of DNA called the tDNA or the transfer DNA, which is the region that actually comes out of the Agrobacterium Thai plasmid region and gets integrated into the nuclear genome randomly, semi-randomly actually. It gets integrated into the nuclear genome semi-randomly. And talking about this Thai plasmid, these are huge plasmids of more than about 200 kilobases. And about 10% of the plasmid region, is transferred to the plant cell after infection. And this region of the tDNA, I mean, this is how the entire evolution has gone. And that now once this tDNA region is transferred from the bacterial Thai plasmid to the plant cell, it starts utilizing the plant machinery to synthesize the genes required for its growth actually. All the enzymes required in growth of the agrobacterium or required for the nutritioning the agrobacterium is already present in the Thai plasmid as well as in the tDNA. So the region that gets integrated into the plant genome, the tDNA region, it starts using plant machinery to start getting things actually. Now, there are a unique amino acid called opine, which is being synthesized by this agrobacterium. It is part of this Thai plasmid and the tDNA region. So there is a gene present called on the Thai plasmid, which is required for opine synthesis, and there is a gene for opine catabolism. Opine acts as the carbon and nitrogen source for agrobacterium. So now you start realizing that why this Thai plasmid and this tDNA has this ability of growing on its own, or rather helping the agrobacterium to grow on its own and develop into a tumor. So because the gene required for, for this opine synthesis and catabolism is present in Thai plasmid and tDNA actually. And this opine acts as the carbon and nitrogen source for the agrobacterium to keep growing. So there are different kinds of opines called octopine and nopalene from arginine, and agropine is from glutamate. And each agrobacterium strain have the opine catabolism gene, the specific kind of opine that it is synthesizing. So whatever opine that is being synthesized by the agrobacterium, the same agrobacterium has the gene for catabolizing it. This means that only a particular kind of a strain can grow in this vicinity. This is like survival of the fittest. So because of this specificity that is being provided by the opines, it causes a particular agrobacterium strain to grow in the vicinity, and no other agrobacterium strain can infiltrate, because that cannot catabolize the particular opine that is being synthesized. So this is part of the part of the survival of the fittest mechanism, that it does not let anything else grow in the vicinity. Now this is just a chemical reaction to show that how different amino acids like arginine and lysine or octapine can be synthesized from the different amino acids actually. Now coming to the tDNA region, we were talking about the Thai plasmid. Coming to the tDNA region, the region that moves from the bacterial Thai plasmid to the plant genome is known as the tDNA region, and it is actually flanked by two segments of DNA, or two signatures of DNA known as LB and RB, which is left border and right border. So anything that is between left border and right border comes out of the bacterial Thai plasmid and gets integrated into the plant genome. So in case of this virulent strain of Agrobacterium tumefaciens, we have the OX-A and OX-B, which are the auxin biosynthetic genes, we have the cytokinin biosynthetic gene, and we have the opine synthesizing gene actually. So these are all the key genes. So as you see, that all the key genes that are required for the agrobacterium to grow and multiply is already present in the tDNA. So apart from some of the other genes which are present in the Thai plasmid. So this opine will act as the carbon and nitrogen source, and then you have the auxin and cytokinin biosynthetic genes. So this leads to the rapid amount or a huge amount of auxin and cytokinin production in the tumor infected, in the agrobacterium infected cells. So, and that's how the tumor starts developing, because there is rapid expression of the, or rapid biosynthesis of auxin and cytokinin, leading to callus slice formation, which looks like a tumorous growth, an uncontrolled tumorous growth from outside. Now we come to the Vir genes. All this process of tDNA transfer does not happen on its own actually. It requires a series of genes that facilitate the process, and this series of genes are known as the Vir genes or the virulence genes. Now there are about 7 or 8 of them, the Vir genes like A, B, C, D, E, F, G7. So all these Vir genes, now you start like walking backwards, you start connecting things backwards. I said whenever there is a wound in the plant, acetylseringione is secreted out into the rhizosphere. Now this acetylserin, and then the agrobacterium comes and infects the plant actually. So just start connecting the links. So this, whenever this acetylseringione is secreted, which is a flavonoid, Vir genes are activated. All these Vir genes are activated, and they are released. This acetylseringione is being released by the wounded plant cells, as I said, and the Vir genes are activated. And these are present about 30 kb of Thai plasmid. It spans about 30 kb of Thai plasmid. I'm sorry, the internet got snapped actually, but I will continue from what we were talking about. So just start, I was saying that let's start connecting backwards, and you will find that we talked about the acetylseringione being released by the wounded plants actually. And what happens, and once this acetylseringione is being released, we talked about the chemotactic response, and the bacteria starts getting attracted, and then it infects the plant. So what actually happens that is this acetylseringione leads to the activation of the different Vir genes, and these Vir genes are present onto the Thai plasmid. So this acetylseringione leads to the activation of the Vir genes, and these Vir genes are responsible for the infection and then subsequent transfer of the tDNA from the Thai plasmid to the plant genome. So different Vir genes have different functions. This transports, the Vir A transports acetylseringione into bacterium. It activates the Vir G by phosphorylation. Now Vir G promotes the transcription of other Vir genes. So you see how a cascade starts the moment acetylseringione is being synthesized and secreted out. Then Vir D2 is an endonuclease. It integrates and cuts tDNA at the border, but only at one strand. And then Vir E2 binds to the single-stranded tDNA, and then can form a channel in artificial membranes. It forms a channel through which tDNA moves actually. Vir E1 acts as a chaperone for Vir E2. And then Vir D2 and Vir E2, they have nuclear localization signals. It gets the tDNA to the plant cell, to the nucleus of the plant cell. And then Vir B is an operon for 11 proteins, get tDNA through the bacterial membranes. So all these different Virs, they have a specific role to play, and all these Virs, I mean, the first Vir to be activated is Vir A, which gets activated by acetylseringione. And once that gets activated, it leads to a cascade of reactions to be taken place by different Virs actually, or virulence genes. We call them virulence genes. Yeah. So this is, we talked about the Thai plasmid. We talked about how Thai plasmid have all the components, the different Vir genes that actually leads to the infection. So this is part of the, I mean, it was actually natural existing virulence mechanism or infection mechanism by the Agrobacterium. Just remember that first. I mean, how it started being utilized for plant transformation is a different part of the story, and we'll come to that later. But for now, just remember that this is a natural existing virulence mechanism of the Agrobacterium. So you see this Thai plasmid. This Thai plasmid has the vir genes, which is spread over about 30 kb of region. You have the origin of replication start site. You have the opine catabolism genes present here. And apart from these three, these are the major components of the Thai plasmid. And inside the tDNA, you have the left border, right border, as I mentioned, LB and RB. You have the auxin biosynthetic gene, the cytokinin biosynthetic gene, and the opine biosynthetic gene. So the opine biosynthetic gene is present inside the tDNA. So once this tDNA gets inserted into the plant genome, this opine can keeps getting synthesized. So see, just look at the evolutionary mechanism or the evolutionary process, that how nature has devised it, so that if the opine biosynthetic gene would have been outside the tDNA, it was the responsibility of the bacteria to keep expressing the opine. But in this case, what has happened actually, because the opine biosynthetic gene is present inside the tDNA, now it becomes a responsibility of the plant. Once it gets integrated, now it becomes a responsibility of the plant to keep synthesizing the opine. Now what bacteria all has to do is to keep catabolizing it and keep using it as a carbon and nitrogen source. So just look at the beauty of this evolutionary process. And to sum it up and to show that how it actually happens, we talked about the different components. Now this is the actual process. I mean, this one figure is to summarize the entire tDNA or the entire Agrobacterium mediated infection process. This sums it up all actually. So I'll just go through the different steps as they are labeled, but you see that this is how it is happening. So this is the wounded plant cell that produces acetosyringone. The very first step that happens is a wound, and this is secreting acetosyringone. Now this acetosyringone activates the vir genes, the vir A first, and then subsequent virs are activated. So the vir protein synthesize single-stranded tDNA. This is the Thai plasmid present inside the Agrobacterium. Thai plasmid having the tDNA, and the one of the vir genes causes a single-stranded tDNA to be formed, which is an intermediate. Now after the third step over here, we will go to the fourth step. Now this with the help of different vir genes, you see, with the help of different vir proteins, which is acting as a chaperone, the vir E2 and all that, which is forming a membrane like a structure, the pilus membrane like a structure. The tDNA is transferred and it gets integrated into, it goes into the nuclear pore, and it gets integrated into the plant genome. It is integrated at random site in the host chromosome or into the plant genome. Now, once this tDNA gets integrated, it starts synthesizing auxin, and then it starts synthesizing biosynthesis. Now, auxin and cytokinin biosynthesis promotes tumor formation. So this is starting to, I mean, this is the generation of the tumor because of the excessive amount of auxin and cytokinin. Tumor formation in Agrobacterium infected plant cells, and then we have the synthesis of opine, which causes the Agrobacterium to keep multiplying in the vicinity. And that's how the infection keeps persisting. So this is the entire mechanism of tDNA transfer and how tDNA transfer is actually causing the infection to go on, the Agrobacterium infection to go on. And this is again the same thing, just a different way of presentation. Here I'm just trying to show that the excised tDNA, some of the vir genes or the vir proteins, they form a pilus-like structure, and they act as chaperones. The vir proteins act as chaperones, which helps the excised tDNA to get to move through this pilus-like structure and starts getting integrated into the plant genome. Now, what are the pros and cons? Like people have found that the monocots, some of the monocots, they do not produce acetosyringone in response to wounding. So acetosyringones are not that prone to Agrobacterium infection, but yes, it is used for transformation purposes by making some modifications. But what is important to see, what people now we are moving that how this infection strategy or virulence strategy is being started being utilized for plant transformation. So what people realized that you put in anything between the LB and RB of tDNA and it will get integrated into the plant cell. I mean, that's what caught people's attention actually. That's what caught scientists' attention. That you put anything between, it does not have to be exactly auxin biosynthesis and cytokinin biosynthesis or opine biosynthesis gene. You just put in anything and it will be transferred to the plant cell. So they asked the next big question. Can agrobacterium be used for engineering plants? Can they put in a foreign gene into the plant to engineer it? But they realized that there were two very critical problems, actually, very big problems. The first one, that the Thai plasmid, the size was huge actually, and it was very difficult to manipulate. Because it was about 200 kb, so it was very difficult to manipulate. And once the tumors, the opine, if you look into some of the literature, the opines that are being synthesized, they lower the transformation efficiency. And once you have tumor formation, you cannot regenerate plants from those tumors actually. So they had this problem. So some people thought that the tumor can grow itself, it is a callus, why not regenerate the plant from there? But they find that that cannot happen actually. And the first major thing was the Thai plasmid was huge and it was very difficult to manage. So then they devised a binary vector system to rescue or to overcome these problems. And this is where the agrobacterium mediated plant transformation actually got initiated. So I think for this particular lecture, I will stop over here because this thing is also coming to the end. The Zoom session is also coming to the end. So I will stop over here and I will have another video prepared which will take you through to the further slides of how this plant transformation takes place and how it is being used in current day modern plant biotechnology. And of course, some of the aspects related to the promoters, the reporter genes, the direct gene transfer, all those will come into the later slides. Thank you for now.